Assalamualaikum and good day everyone. We are from Group 2 will present to you guys the chromosome aberration which is Jacob syndrome. These are our group members. In this presentation, we will discuss about the introduction of Jacob syndromes, etiology, clinical manifestations, diagnostic testing, complications and treatment and management.
This is a virtual karyogram and the picture on the left is a normal one and the picture on the right is the abnormal one. And this is physical karyogram, the picture on the left is the normal one and the picture on the right is the abnormal one.
This is our arranged karyogram and we found an extra Y chromosome which confirmed the diagnosis of Jacob's or XYY syndrome. Firstly is the introduction. Jacob syndromes refer to male presence with extra Y chromosome which produces a 47 XYY karyotype. It is a rare genetic disorder that occurs in about 1 out of 1,000 male children. Sometimes it is also known as super male disease, as usually men with this syndrome were thought to be overly aggressive and lacking in empathy. This type of genetic disorder belongs to a group conditions known as sex chromosome trisomies. Next is the clinical manifestation. For the clinical manifestation, we can notice physical and mental difference in comparison with normal male with XY karyotype. However, signs and symptoms of XYY syndrome can vary greatly from individual to individual. Some boys have no obvious signs while others have mild symptoms. Occasionally, the disorder causes significant problems. Boys with XYY syndrome may have some or all of these physical symptoms to some degree. Taller than average height, low muscle tone or muscle weakness, which is called as hypotonia, very curved pinky finger or clinodactyly, widely spaced eyes known as hyperthyroidism, microcephaly, severe cystic acne during adolescence. As mentioned previously, some XYY males also develop mental problems such concerning their social, language and learning skills. They also can have problems with reading and understanding math and may have mild delays with coordination. In summary, the neurological problems in XYY males include developmental delay, speech and language delay, learning disabilities, behavioral or social problems such as explosive temper, hyperactivity and impulsivity, but with normal fertility and sexual development.
And next we proceed with complication. Apart from the signs and symptoms manifest in Jacob syndrome patient, there are few complications or increased risk of comorbid disease in this patient. This includes the first one is increased incidence of asthma, seizure disorder, dyslexia and tremors. The second one would be infertility due to oligospermia or sperm chromosomal abnormalities. And the third one would be genitourinary problems such as microphilus, hypoplastic scrotum, and cryptorchidism, hypopardias. And the fourth one would be increased testosterone level related to increased risk of aggressive behavior. And the last but not least is learning disabilities, ADHD, autism spectrum disorder, and speech difficulties. And next, we continue with the etiology. Jacob syndrome is not an inherited condition. Male with XYY syndrome have one extra Y chromosome due to either one condition. The first one would be the partenomyosis II non-disjunction, which occur due to failure of sister chromatid to separate in NFase II during the formation of sperm cells. The second one would be postzygotic mitotic non-disjunction, an error that occurs during division of sex chromosome in the zygote after fertilization. It is unknown if there are any parental causes for this mutation. Next, we carry on with diagnostic testing. The median age of diagnosis Jacob syndrome is approximately 17 years of age, in which patient presenting due to infertility concern. Two diagnostic measures can be carried out. The first one is prenatal testing that will be done before delivery, and the second one is postnatal testing. Few tests of prenatal testing include cell-free fetal DNA, amniocentesis, and chorionic villus sampling (CVS). The cell-free fetal DNA tests are the non-invasive prenatal testing which diagnose fetal DNA that circulate freely in maternal blood. Meanwhile, the amniocentesis and chorionic villus sampling are invasive testing, which analyze amniotic fluid and tissue sample from the placenta respectively. Both samples would be tested further for chromosomal study. For the postnatal testing, karyotype analysis from the patient's peripheral blood karyotype analysis is the gold standard for Jacobs syndrome. Next, we keep on with the treatment and management. There is no cure for Jacobs syndrome, but treatment can help with specific symptoms. Treatment for Jacobs syndrome is supportive and based on the signs and symptoms shown by the patient, which may include speech therapy, occupational therapy may be needed if hypotonia is present, appropriate medical treatment for comorbid conditions such as asthma and seizure disorder, and last but not least is correct hormonal imbalance of the patient. And this is our references, and this brings us to the end. Thank you so much for the attention.
Now we will present our cytology laboratory report.
For the purpose of this lab, we are going to use two types of staining. The first one is MGG, which is May-Granville-Jamesa stain, and the second one is Papa-Nicolau stain, which is also known as PAP stain. This is the procedure and interpretation of MGG stain. Here is the picture of the MGG staining process. The second stain we use is the PAP stain, which consists of 5 dyes, namely hematoxylin, orange G6 counter stain, eosin azure counter stain, eosin Y, and also light green SF yellowish. Here is the procedure and interpretations of PAP stain.
Next, we proceed with the lab technique. First technique is for the vaginal smear and swab. We use the flushing technique to collect sample from the vaginal canal. For the staining of the sample, we can use either the PAP stain or the MGG stain.
For the bulk swab technique, firstly open the swab package, then place the swab tip into the mouth and rub the side of the cheek about 6 times. Then take off the swab from the mouth and directly smear the sample on the slide. Staining that we use in this technique is MGG stain.
Next technique is scraping. First, we collect skin by scraping and place them on the center of the slide. Then, we put another clean slide face down on the slide with specimen and let the specimen slowly spread out between the slide. After a few seconds, we separate the two slides and the slide is immediately fixed in the 95% ethanol.
Next is organ impression technique. First up, using a surgical blade to cut the tissue into two and then wipe the fresh edge to eliminate extra fluid and blood, and make 3 impression smear on glass slide by gently touching tissue's fresh edge to it. The process is then duplicated and then directly proceed with PAP staining for the first slide and then allow to air dry and proceed with MGG staining for the second slide.
For the urine cytology technique, clean cache urine is collected. Then the urine is centrifuged. Supernatant is discarded and the remaining pellet is used for smearing on the glass slide. The smear was left for air dry. Then the glass slide is stained with MGG or PAP stain. After the staining process is completed, the slide is ready for screening by cytology.
This is fine needle aspiration procedure. A needle is attached to a string and inserted into the organ of interest. Apply suction for a few times and release the suction, then we trunk the needle. After that, disconnect the syringe from the needle and fill it with air by pulling the plunger. Then we attach it back to the needle. Eject the sample onto the slide and finally smear the aspirates and stain with PAP and MGG staining.
For vaginal sampling using pap smear, we also superficial squamous cell that's shown by the arrowhead. We can also see an intermediate squamous cell that's shown by an arrow. Extra stage in this sampling shows a pro-estrous smear that consists of predominance of nucleated epithelial cells. As for cell in reference, we can see nucleated cells with pink cytoplasm, densely packed nucleated cell with dark blue to purple staining granulated nuclei. While for the estrous stage, it is pro-estrous smear densely packed nucleated epithelial cell. Next, we go to vaginal sampling that's stained using MGG. We observe a larger nucleus, 8 micrometer in diameter and polygonal shape with transparent pink cytoplasm. while in cell reference, it has predominance of nucleated epithelial cells that have lightly basophilic fibrillar cytoplasm and a single, relatively small, central round nucleus that's shown by black arrow.
To obtain the buccal sample, we use swab technique and visualize using MGG staining. So what we can see from the buccal swab done in the lab under 40 magnifications are presence of squamous epithelial cell, polygonal shape, small round nuclei, and centrally located nuclei. And for the buccal swab reference, what we can see is presence of normal squamous epithelial cell, polygonal shape, the nuclei are round or oval shape, normal nuclear cytoplasmic ratio, and centrally located nuclei.
For skin scraping sampling, we observe an isolated cell that look like epithelial cell with MGG staining. However, we need to troubleshoot for further better observation. While troubleshooting, we found that proper sampling technique need to be applied to obtain sufficient sample. Inadequacy of cells due to poor sampling technique make it hard to observe. Stain is too weak due to less time of staining. For reference, we compared it with epithelial cell from buccal swab, where the shape is likely the same, polygonal shapes, and has low nuclear cytoplasmic ratio.
The impression smear technique is applied to the liver samples of the rat for liver cytology, and MGG stain is applied onto the slice. The reference cytology slice shows normal hepatocytes, which are presented as sheets of large polygonal cells with an abundant granular cytoplasm and possesses rounded nuclei with prominent nuclei. The laboratory results are like the reference result. The overlapping cell calls for an improvement in the technique of impression smearing.
We use dabbing technique to obtain the sample from kidney and stain it with MGG staining. Under 40 times magnification, we observe dispersed and small cluster of cells with small nuclei, pink cytoplasm, and normal nuclear cytoplasm ratio. As for the cells in reference, it appears as dispersed cell with small and spherical nuclei, pink granular cytoplasm, and normal nuclear cytoplasm ratio. We need to troubleshoot by removing exudates prior to impression smear to prevent significant blood contamination.
To obtain spleen sample, we use Mia technique and visualize it using MGG staining. And under 40 magnification, we can see the erythroid precursor. No other cells are clearly visible, predominantly erythroid precursor. And for troubleshooting, try on the other techniques such as brushing the superficial part of the organ instead of direct smear on the slide to obtain more cell. And the reference for spleen, we can see the normal lymphoid cell, segmented neutrophils.
For lung sampling in 40 times magnification, we can observe pulmonary alveolar macrophage, eccentric nucleus, abundant and foamy cytoplasm. In cell reference, it shows there is presence of pulmonary alveolar macrophage, eccentric nucleus, abundant and foamy cytoplasm, while the other picture of the cells shows there is ciliated epithelial cells, eccentric nucleus, oval nuclei, finely stippled chromatin, and cilia project from apical surface. To compare between our observation and cell reference, the cell reference shows more clear picture. To troubleshoot this problem, we need to use 100 times magnification for a better view. In heart sampling using MGG stain, we observe a neutrophils, cardiomyocyte, one elongated nucleus that lies in the center, while in a reference cell there is cardiomyocyte that shown in red circle, intramuscular capillary that shows by green arrow, cardiac muscle cell nucleus in the yellow circle. For a better view, we need to use 100 times magnification.
Next is the observation of ovary samples obtained from rat dissecant and applied onto slides with fine needle aspiration technique. In the reference slide, a well-differentiated endometrial ovarian adenocarcinoma is observed, with round to oval and a hyperchromatic and eccentric nucleus with finely granular irregular distributed chromatin with prominent nuclei is observed. The cytoplasm is irregular and scanty. In the laboratory slides, large ovarian surface cells with abundant cytoplasm and pale staining nucleus with prominent nuclei is observed. Poor sampling technique and sample inadequacy may cause the slides to be presented in a slightly desensitized pattern. Next, the impression smear technique is applied to the urine samples of humans for urine cytology and PAP staining as well as MGG staining. The reference cytology slides as well as our own laboratory results show the presence of urine squamous epithelial cells, which includes superficial cells and intermediate cells. The laboratory results are like the reference result. However, the MGG staining does not produce a clear picture of the cytology slide when compared to PAP staining.
The liver sample is obtained by fine needle aspiration technique and being stained with MGG staining. Under 40-time magnification, we can observe hepatocyte with small nucleolus and cytoplasm with inconspicuous cell borders. Liver sample under 400 times magnification showed hepatocyte with single nucleolus, small amount of nucleolus cytoplasm, and cells are found in cluster in hepatocellular carcinoma. We need to troubleshoot in order to obtain sample with more skilled technique to get adequate sample.
Uterus sample obtained by scraping technique and using MGG staining. For the lab sample, we can see isolated cell, scant cytoplasm, nucleus is either equal or smaller than intermediate cell nucleus. For troubleshooting, use other technique or improve skill of transferring sample to slide to obtain adequate cells. And for reference sample of the uterus, we can see the elongated cytoplasm and the presence of ciliated, which is indicated by the black arrow, and the non-ciliated, which is indicated by the white arrow cells. And this is our reference. That's all from us. Thank you so much.